Good afternoon, everybody. Welcome to today's Wednesday afternoon lecture. I'd like to remind everybody that there will be an FAS-sponsored reception in the library immediately following the lecture. We hope that you will join us. It is my pleasure today, on behalf of the Fellows Committee, to introduce Ricardo Dolmetsch, our Wednesday afternoon lecture speaker. Dr. Dolmetsch attained his Bachelor of Science with honors from Brown University. He then went on to get his PhD in neuroscience from Stanford. He performed his postdoctoral studies in the lab of Dr. Michael Greenberg at Harvard University School of Medicine. He has served as the Senior Director of the Allen Institute for Brain Research in Seattle, Washington, and an Associate Professor in the Department of Neurobiology at Stanford University School of Medicine. He joined Novartis in June 2013 and is currently serving as the Global Head of Neuroscience in the Novartis Biomedical Research Institutes. He is charged with the formidable task of leveraging advances in human genetics to model and treat neuropsychiatric and neurodegenerative diseases. Reports suggest that he was curious and interested in science even as a child. One report indicates that he may have sacrificed his father's prized stereo amplifier to test stretch receptors in hand-caught cockroaches when he was young. That sacrifice has obviously paid off. He has made some major research contributions, including the development of induced pluripotent stem cell-derived neurons to model cellular and cardiac phenotypes associated with Timothy syndrome, which is a rare disease characterized by cardiac arrhythmias and the features of autism. He has also developed novel technologies to facilitate the study of neuroscience, including systems for light-activated protein dimerization and gene expression in live cells and animals. And he has advanced the understanding of the cellular and molecular basis of autism and other developmental disorders, including the role of calcium channel signaling in the developing brain. He has also self-acknowledged some pioneering work in the utilization of the foam mattress to take naps between experiments. He is the recipient of numerous awards, including the McKnight Technological Innovations in Neuroscience Award, Burroughs Wellcome Career Award, the Society for Neuroscience Young Investigator Award, the prestigious NIH Pioneers Award, and the Stanford Medical School Distinguished Teacher Award. That's just to name a few. He's served on many committees, advisory boards, and study sections, as well as being an editor for the Faculty of 1000, and serving on the editorial board of Cellular and Molecular Neuroscience. He has expressed the admirable desire to one day open a research and educational foundation in his country of origin, Colombia. And he has also received press coverage for campaigns to abolish lint and to provide a goldfish to every lab member that he has. This is on the web, folks. I can attest from lunch that he is also amusing, engaging, and has a great sense of humor, and I'm not even looking for a job in neuroscience. Please join me in welcoming Dr. Ricardo Dolmetsch to give today's Wednesday afternoon lectures.
Boy, the internet is a terrible thing. You do something when you're 25 and it follows you forever. Today I'm going to tell you a little bit about the work we have been doing in my lab over the last decade, but also about some of the work we've been doing at Novartis over the last 2 years to try and develop new treatments for diseases of the nervous system. And I thought that before I start, I would tell you just a little bit about myself, because this, I think, will help you understand why it is that I do what I do. So I think there are two things you should know. One is that I was, a very basic neurobiologist, very interested in understanding how ion channels contributed to the development of the brain, and then my son was diagnosed with autism, and with a whole bunch of other things. It turns out that this says a lot about psychiatry. He has like 5 different diagnoses, and he's comparatively high functioning. But this really changed the direction of my career, and I became very interested in the underlying cellular and molecular basis of autism and other neurodevelopmental diseases. So that's one piece of evidence. The other thing you should know is that I'm originally from Colombia, and so what this means is that my mom calls me twice a week, and I provide some of the world's most expensive computer advice. And so she calls me, and she doesn't fully understand what I do, but she does feel the need to give me motherly advice every time I call. And so not so long ago, a couple of years ago, she called me, and she knows that I work on autism. And so she said to me, I'm amazed at how much progress you're making in autism. Every day you discover something else that causes it. And, it's sort of, damning with faint praise. It didn't feel like, it felt like failure. And so, in part because of that, because I felt that, we weren't really moving towards helping anybody, I decided to move to a company where where we could perhaps develop some treatments for some of these neurological and psychiatric diseases. And, our goal at Novartis is to develop medicines that will reduce human suffering, and specifically by treating diseases of the nervous system. And in fact, there is a great deal of suffering to go around. Major depression, for example, there are 50,000 deaths in Europe every year from suicide, and that number hasn't changed in a long time. Alzheimer's disease. There will be 130 million people with Alzheimer's in only a few decades. And if we don't come up with good ways of treating AD, we risk bankrupting our medical systems. 1% of the population has schizophrenia, and most of these people are in jails and in homeless shelters. That's how we take care of our schizophrenics. Addiction. There are 15 million people in Europe alone with addiction. We don't have any treatments for that. And then finally, of course, what's closest to my heart, sort of autism. So the number, I think, is 1 in 66, but some fraction of those kids actually exit the diagnosis. About 1 in 100 will require lifelong care. So the needs are huge. And, unfortunately, we haven't been very effective at coming up with treatments. Now, it wasn't always this way, right? Actually, let me just exit here 1 second. Something has happened here. Sorry about this. Yes. There's nothing so terrible as realizing that you have the wrong slide presentation, especially if you work for a company. There are a whole bunch of Swiss people who would go nuts if I presented that other slide presentation. So, as I was saying, in fact, we haven't always been unsuccessful, but the last 20 or 30 years have been really hard. And, at Novartis, we have a fairly proud history, right? So this was the place where, and the predecessor companies, which were Sandoz and Ciba, these were the places where, we developed a lot of the armamentarium that is used in sort of modern neurology and psychiatry. So everything from Ritalin to carbamazepine to all the way to Gilenya, which is the first oral treatment for multiple sclerosis. So you'd think we would be in this area, right? You'd think all of pharma would be in this area. I mean, look at this, right? This, if not only does this result in helping lots of people, you'd think this would make lots of money. And the answer is it turns out that no, everybody's leaving neuroscience. And people are leaving neuroscience because of this. So this is from a review by Michael Ringel and Ulrike Schultz, but it illustrates, it's from a sort of overview of what it is that makes a drug company successful. And it turns out that there are a lot of things that add to the positive impact. In fact, the disease area doesn't much matter unless it happens to be neuroscience. It turns out that if you're a drug company, neuroscience is negatively correlated with your success. And so companies have voted with their feet, and they've started exiting the field. And the question is why? Why is it that things have been so rough in recent years? Why is it that the failure rate has been so high? And I think, and also, of course, what is it? What can we do to actually change this? And you know, I think there are at least 4 things that we have going against us. So you've all probably seen something like this. This is a process of drug discovery, right? You start with disease and target selection, and then you model things preclinically, you screen, you optimize, and then you develop whatever it is that you optimize clinically. And it turns out that every single one of these steps is really hard in neurobiology. So let's start from the very beginning, right? So in general, in neuroscience, we have selected blockbuster indications that we didn't understand very well. And I think we were fooled a little bit by early successes in, you know, for example, with antipsychotics. But almost all of those were actually serendipitous. And in fact, when we set out to develop specific therapies aimed at specific targets, we selected diseases like depression, which is not a single disease, and schizophrenia, which is not a single disease. And so because we didn't understand the underlying pinnings of the disease very well, we were stuck with symptomatic treatment, and we were developing those symptomatic treatments using a set of assays that we had essentially optimized for specific sets of pathways. So, you know, we need to select better diseases. We need to select diseases where we have some chance of succeeding. Our preclinical models suck. They're just not very predictive. You know, for a long time, we were developing antidepressants based on their ability to get mice to swim in buckets. And it turned out that, you know, that worked for some of the existing antidepressants. But after a while, we couldn't make drugs that were any better because we had sort of maximized that assay. And of course, many of the things that make mice swim longer in buckets do not actually translate to humans. You know, neuroscience drugs have to be really safe. We're mostly giving them to people who are comparatively healthy, and we're often giving them these drugs for the rest of their lives. Now, in addition to this, they have to get across the blood-brain barrier. You know, I sometimes tell people I'm sort of in awe and, you know, somewhat jealous of my colleagues in oncology because, you know, if you're going to develop a drug in oncology, you just have to make sure it doesn't dissolve the mouse. But that's not true at all in neuroscience. We've got to make drugs that are very, very safe, and that takes a lot of time. And then finally, of course, our clinical development path is really hard, and there are at least 2 reasons for that. The first is that our patient populations are really heterogeneous. So we are trying to go into people who we think have a single disease, but it turns out that they have a bunch of different diseases. Now, even worse than that, we have a whole set of soft endpoints. There are, you know, we're always measuring things like mood and motivation and attention and memory, and it turns out that that is all terrifically variable. So we don't have really good ways of trying to measure whether our drugs work at all. So if we're going to have any success at all, we have to improve in all of these areas. So I'm going to tell you a little bit about how we're trying to do that. So let me just start with, you know, how to select targets. So our goal at Novartis is to try and tie every drug development program to a patient and to a patient population. So we're trying to develop things with the end in mind. And there are really 2 ways you can do that. So approach number 1 is essentially illustrated here. So it's to start to rely on serendipity. And this has been the history of success in neuroscience. So many of you may know the story of chlorpromazine. So chlorpromazine was the first antipsychotic. It was never developed as an antipsychotic. It was developed as an antihistamine. And then this man, Henri Labourite, noticed that it was also a sedative. And so he was using it in patients before surgery. And he had 2 friends, Pierre Dennecker and Jean Delay, who were psychiatrists at the same hospital in Paris. And he noticed that the schizophrenic patients were very agitated. And so he suggested that they ought to try this drug, which he loved. And he discovered, somewhat to his, they discovered somewhat to their surprise, that it sedated them, but also it reduced some of their psychosis once the sedation wore off. And then what happened after that was sort of interesting. So then this drug was then taken back into animals, and a set of preclinical pharmacological assays were developed around the drug. So it was found, for example, that it antagonized amphetamine. And so the subsequent development of other antipsychotics was essentially a chemical random walk starting with the structure and using pharmacological preclinical assays, like, for example, the fact that this would antagonize amphetamine, to develop new antipsychotics, with the result that essentially all antipsychotics work by antagonizing amphetamines. So that's one approach, and I should say that this is an approach that we need to continue using. We need to take advantage of clinical serendipity to develop new drugs, in part because we need things that work in people. And so there are some opportunities in neuroscience where something that, where a drug has a psychiatric or a neurological effect and it can be improved. And so as a drug company, we need to do that. But that's a very small number of things, and we're very unlikely to make dramatic breakthroughs that way. So the other approach is sort of illustrated here, and that's to take advantage of the revolution in human genetics. And you're all familiar with this, which is the dramatic decrease in the cost of sequencing. The cost of sequencing a genome has fallen by about 1 million-fold, and this, of course, has made all kinds of experiments possible that were not possible before, and has led to the identification of a whole series of genes that either predispose you to disease or, frankly, cause specific diseases. And sort of the way the landscape at the moment in neuroscience, it looks sort of like this, right? So if you have a graph of the effect size and the allele frequency, there are essentially 3 classes of mutations. There are these rare Mendelian mutations that have a high effect, so they're pretty penetrant. So every time you have it, you have some disease, but they're very rare. There are sort of low frequency copy number variants. These are more common, but they're somewhat less penetrant. And so the actual phenotype associated with this microduplication or microdeletion is somewhat variable across a population with that deletion. And then finally, at the other end, there are these common single nucleotide polymorphisms. These are the things that are identified by genome-wide association studies. And so the question is, how can we convert this kind of information into any sort of drug discovery program? Let me just say, so the question then is, how does this actually contribute to disease and to changes in behavior? And the model sort of looks like this, right? So we all have a genetic background, right? These all single nucleotide polymorphisms are what make me, a short Colombian guy. But then superimposed on this are these copy number variants and rare mutations, and together they kind of tilt the balance and cause disease. Now, our challenge is to convert these things into drug targets. And I should say that, of course, it's progressively easier to develop drugs as you move in this direction. So for rare Mendelian disorders, there are some mutations in pathways and in enzymes that are frankly druggable. For low-frequency copy numbers, sometimes we can make a guess as to the gene or genes that cause the disease. But for these common or single nucleotide polymorphisms that in combination cause the vast majority of disease, here we don't really know. And so I'll tell you a little bit about some of the things we're doing to try and develop this kind of genetics into drug discovery programs. Okay, so the second issue is, how are we actually going to do this? So drug development is all about assays. If you, the better your assay, the more it reflects the disease, the better your drug. It's all about being able to optimize something preclinically such that it is likely to work in people. And so to do that, your preclinical assay has to somehow reflect the disease. And so conceptually, right, you could imagine that, we have changes, we have genetic changes that lead to changes in behavior, and conceptually this happens in a fairly straightforward way. So the genes, of course, change the function of cells, and the cells change the function of circuits, and this in turn impacts behavior. Sounds easy. The question is, how do we actually convert this into some sort of an assay that can be used to optimize a series of molecules? Historically, the approach is we would make a mouse. So we would take a human mutation, and we would introduce it into a mouse genetic background. If you're in China, you make it out of snow. And then we would characterize the mouse. And this has been sort of I would say, a sort of a mixed bag. I mean, mouse models have been useful in some areas, and not so useful in others. I'm going to give you an example of a mouse model that we generated in the hope that this would be useful for drug discovery, and how it initially led us astray. So we were interested in this disease, 16p11.2 deletion syndrome, which is rare, but not so rare that you wouldn't be able to find enough people to do a proof of principle clinical trial. And it was initially, this is a copy number deletion, and that it deletes 27 genes on human chromosome 16. Now, fortunately, it is synthetic on mouse chromosome 7. Now, the patients have, it's a highly penetrant mutation in the sense that about 75% of the patients have some sort of a psychiatric diagnosis. But in fact, which psychiatric diagnosis they have is, varies according to different individuals. So the most penetrant diagnosis is obesity, but then, there's, autism, defects in language, intellectual disability, and ADHD. And so, so we decided to make a mouse because we could, in fact, delete all of these genes. And so we generated this mouse, and then we tried to identify some phenotypes. Now, we hoped that some of the phenotypes would be social phenotypes, and so we actually did sort of an interesting experiment. We phenotyped the mouse at a contract research organization. We had a collaboration with Jackie Crawley, and we also phenotyped the mouse at Novartis. And there are a couple of interesting things. First of all, only 2 phenotypes actually reproduced across all 3 sites. So that told us that, in fact, identifying a phenotype was going to be sort of difficult. Now, one of the phenotypes that reproduced was something that we really didn't expect, which was sort of a defect in something called social memory. And this is the way the assay works is sort of illustrated here. So what you have is sort of your mouse, and you're presenting your mouse with a female, and then you're seeing whether the mouse is excited to see the female. And so, the first time this mouse sees the female, he's very excited, and then you remove the female and you wait 10 minutes. And when you do this, the mouse is somewhat less excited. And then you wait another 10 minutes and you present the same female. He's already seen the female. He's significantly less excited. And then as a control, you introduce a brand new female, and he's really excited again. Okay? So the way it works, so we did this experiment on our mouse, right? So this is our wild type mouse, and you introduce a brand new female, and he's very excited to see the new female. Okay, and you do this in the 16p control, and he's also very excited. We were a little disappointed by this. We were hoping that he was going to be antisocial, but it turns out he wasn't. So now we're going to jump to 40 minutes later, and this is what you see here. So now we introduce the wild type, and he's not quite as excited about this female because he's already seen the female before. The 16p mouse, on the other hand, is still very, very enthusiastic. In fact, the person who was doing the experiment in my lab said that this was sort of like the ideal husband. And then you can, of course, do the control and sort of introduce, a new female, and you can see that they're interesting. So now the thing is that this is exactly the opposite of what we expected. Right? What we expected was a mouse that was going to have a social defect because we were trying to make a model for autism. And in fact, if anything, this is a mouse that was particularly attracted to other mice. It had perhaps some memory defects, but subtle ones. And it turned out that, in many ways, and in fact, these mice were unusually thin, the patients were obese. So if you looked at the phenotypes of the mouse, in fact, the mouse was a fairly poor phenocopy of the human mutation. And I would say that this is true for virtually every mouse strain that we've made using a mutation for a psychiatric disease. So I think what we've learned is that, if you're looking at behavior, there's been a lot of divergence over evolution, which means that mice are just not very good models of psychiatric disease. And in fact, they're not very good models of neurodegenerative disease either. But that's what we've been using. Now, the correlation of that is that the further away you get from the original mutation, the less predictive the model becomes. So, for example, if instead of looking at the behavior, instead we looked at the cellular defects, And as I'll tell you in a second, we were able to look at the cellular defects in cells from the patients. Those were somewhat conserved. But as you moved away from the original mutation to something much more complicated like behavior, that was poorly conserved. And so if we had tried to use this as a way of developing a new drug, it would never have translated. And in fact, that's been our observation. Many of the drugs that we've developed using behavior as a readout have just not translated because behavior is very divergent relative to the initial cause, the initial insult. So the other approach is to try and study people. Of course, we can't study human behavior, but we can study human cells. And so, of course, this is not a surprise to oncologists, for example, who have been collecting biopsies and making collections of them for a really long time. And the good news in oncology is that, to a first approximation, a tumor behaves in the lab sort of like it behaves in a human. And so you can use this as a way of screening for a lot of drugs. But of course, the problem we have in neuroscience is we don't really have any way of collecting a biopsy from somebody's brain. So the next approach is to see if we can make a biopsy. And so the approach that we developed now almost a decade ago with help from actually from NIMH was to take advantage of this sort of miraculous discovery by Shinya Yamanaka and his colleagues. And the basic discovery was that you could take somatic cells like skin cells or blood cells that were fully differentiated and specialized for that function. You could introduce these reprogramming factors into the nucleus, and you could then convert those cells into stem cells. And so the reason that was powerful is because we could then sort of sit or stand on the shoulders of giants and use many of the discoveries that had been made in understanding developmental neurobiology to actually convert those pluripotent stem cells into neurons. Now, these two steps, harvesting skin cells from patients and reprogramming them to make stem cells, these have become commodities. So I'm not going to go through the process. Let me just tell you that it's now possible to make hundreds of these lines in an automated way, in a way that is essentially scarless, so you don't actually alter the genome. And we can do this in a very reproducible way, and we've made hundreds and hundreds of these lines. What is not quite so easy yet is to convert these stem cells into neurons and then to use these neurons to see whether we can develop some new drugs. So how to convert stem cells into neurons? And so some of the initial protocols essentially used an approach that tried to recapitulate development. And so the way, for example, that we did it early on is sort of illustrated here. So we would take the iPS cells, we would withdraw a particular growth factor, FGF2. This would generate these embryoid bodies that have, to a first approximation, the 3 germ cell layers. You can take these and then you can plate them, and then they would start to form these things that are called rosettes, but they're very much like the developing neural tube. And we knew this because we could actually measure a bunch of markers that are characteristic of the developing neural tube. You could take these and pick them up and then expand them as these neurospheres. And these neurospheres actually, as I'll show you in a second, have very interesting sort of 3-dimensional structures to them. And then you could, or you can dissociate them and generate these 2-dimensional cultures. And if you waited long enough, you could make, neurons and glial cells. In fact, it's been possible now to use this approach to generate not only planar cells, but also what start to approximate a structure that starts to approximate a little piece of cortex. So this is the work of Sergiu Pașca, who was a former postdoc in my lab and now has his own lab at Stanford. But basically, you can take these neurospheres and you can let them grow for a long time. And if you do this, you generate something that starts to look a little bit like a brain. And in fact, you can actually section something like this, and you can see that there are multiple layers. There are, for example, neurons that have markers for deep cortical layers as well as superficial layers. Now, so it seems then that we can make neurons. So the next question, of course, is are these neurons really neurons? Do they behave like neurons? Is this the kind of thing that we want to use to actually develop a new generation of drugs? And of course, there are many questions, right? You can imagine that there are many possible cell culture artifacts. But we've done a lot of characterization over the years. I'm just going to give you a couple of snippets of how we've characterized these cells. So one of the things we can do, for example, is use either single-cell RNA-seq or single-cell RT-PCR to look at the sets of genes that are expressed by particular sets of cells. And so, for example, this is from a single-cell RT-PCR experiment done with this machine called a Fluidigm machine. And so every cell, so each one of these is a cell, and every cell has a kind of barcode that tells you which genes it expresses. And based on the genes that it's expressing, we can determine whether it's a precursor, whether it's a dorsal precursor or a ventral precursor, whether it's a neuron, if it's an excitatory or inhibitory neuron, which sort of cortical layer it belongs to. And you can do this for tens of thousands of cells. And if you do this, you can see two things. The first is that, using this particular differentiation protocol, and this is at day 90, so this is after 3 months, there are quite a lot of neurons, but there are also quite a lot of precursors. The precursors are mostly dorsal, so it's this part of your brain. There are cells that have markers for all the different cortical layers. The second thing you can see is that there are error bars here, and each one of the error bars corresponds to a different differentiation. And so this was helpful because it told us that, in fact, differentiation was very reproducible, which, of course, is very important if you want to use this as a way of identifying a phenotype associated with a disease. Of course, we wanted to make sure that these cells were functional, and so we could, for example, load them with a calcium indicator. And if you did this, you could see that they had, calcium elevations when they were stimulated electrically. You could actually patch clamp them. Now, to do this, we had to develop a whole new set of markers that would allow us to make sure that we were always patching cells that were actually mature neurons. So this, for example, is a fluorescent gene that is only turned on in mature pyramidal neurons. And so if you patch onto the cell, you see beautiful action potentials. We discovered, for example, that if you just simply cultured these cells, they didn't form synapses very well. But if you cultured them either in an organoid or with glial cells, then they would form beautiful, beautiful synapses. And you could measure synaptic responses. And of course, we measured hundreds and hundreds of these. And you could see, for example, that there were, at different stages of maturity. And then finally, more recently, we've taken these cells and we've reintroduced them into mice. Now, the reason we've reintroduced them into mice is not so much because we think that they're going to recapitulate the disease in the mouse, but because we have been able to identify, as I'll tell you in 1 second, cellular phenotypes. And if you put them back into a mouse and you give the mouse a drug, then you can test to see whether your drug is getting into the brain and whether it's actually changing the cellular phenotypes of the human cells in the brain of the mouse. And so this has turned out to be sort of a useful model for trying to test whether a particular compound gets to the right place and does the right things. Okay, now one of the advantages of working in pharma is that we have the expertise and the resources to be able to do this on a much larger scale. So one of the problems that we found when we first started doing this was that it turned out that if you wanted to differentiate 2 or 3 or 4 lines, it took one person full-time for 3 months to differentiate 3 or 4 lines. It was just an enormous amount of cell culture, and this meant that every experiment was just not terribly reproducible. So we have invested heavily in developing the technology to actually automate this process. So this is, for example, one of our robots that allows us to do essentially the whole feeding and differentiation process automatically. And so we can do, around 100 cell lines at once. And so this is helpful because it allows us to, for example, differentiate the same cell line multiple times, identify the noise, and of course, if your goal is to develop a really robust assay, you need to be able to do that. Okay, so now let me tell you a little bit about our sort of disease strategy at Novartis before I go into some of the projects we're working on. So, in Novartis broadly, we're interested in 3 areas of neuroscience. We're interested in multiple sclerosis, neurodegeneration, and psychiatry and neurodevelopment. Now, for multiple sclerosis, almost all the work comes from our immunology and transplantation group. So I'm not going to tell you very much about this area of our work. instead, I'm going to focus on these two, neurodegeneration and psychiatry and neurodevelopment. So let's start with neurodegeneration. Of course, one of the areas that we're interested in is Alzheimer's disease. I would be the world's worst head of neuroscience if I didn't think that Alzheimer's disease was a big problem. This is you know, a sort of approximation of what cognitive decline looks like as you age and if you get AD. And there are essentially three kinds of therapies that we are working on. So the first one is, of course, we're working on ways of trying to inhibit the accumulation of A-beta peptide. And so work from a number of groups, but primarily from a consortium. The ADNI consortium suggests that A-beta, which is this fragment of the APP protein, accumulates in the brain of people who are going to get AD. And this starts, you know, when people are in their late 40s and accumulates over time. And the genetic evidence that this is important in AD is very strong. So there are people, for example, that have mutations in APP that confer susceptibility, get early onset Alzheimer's. There are people who have mutations in the enzymes that produce this peptide, and they get Alzheimer's. And then more interestingly, there are people who have mutations in the precursor, right, in APP, in isolin in particular, that are protected from the disease. And so we have developed a very safe and very potent inhibitor of one of the enzymes that produces this A-beta monitor base. But of course we were late to the game. And we were late to the game in part because you know, neuroscience at Novartis was shut down for a bunch of years. So the question is, you know, given that we had this really great compound that was at least partly developed with the knowledge of all the flaws in everybody else's compound, what were we going to do? And so we thought about it, and then we thought, well, you know, given what we know about the biology, perhaps we should do something that pharmaceutical companies hate doing, which is to do actually a prevention trial. And so we partnered with the Banner Alzheimer's Institute and with NIH to do a prevention trial. And so actually this prevention trial will test both our BACE inhibitor as well as a vaccine that generates antibodies against these A-beta monomers. It's a 5-year trial, but of course, you know, if you're going to do a prevention trial, you have to be fairly certain that people are going to get the disease. So this is a trial in people who have 2 copies of the APOE4 allele, which we know confers in fact, about 90% of these individuals will develop AD. And so this is sort of our approach to trying to develop a drug for this stage of Alzheimer's. We're fairly certain that this is not the only thing that's going on. In fact, the evidence suggests that, for example, even though this is important in causing the disease, the amount of A-beta does not correlate very well with cognitive decline. There are some people who die with lots of A-beta in their brain and they're cognitively intact. On the other hand, tau is very well correlated with cognitive decline. And so tau aggregates and tangles are another pathophysiological characteristic of AD, and so we're developing some drugs that interfere with the production and secretion of tau. And I should just say, tau is very unusual. It's an intracellular protein that seems to be secreted and somehow seems to go from across cells. Now, the final part of this is that there seems to be activation of an inflammatory cascade and the activation of these phagocytic cells in the brain, the microglia, and it seems that this is actually important for the neurodegenerative process. And of course, we'd like to have a drug that we can use when people actually show up to their neurologist, not in preparation for the disease, and so we're working on this as well. Now, in addition to our interest in AD, we're also interested in these orphan neurodegenerative diseases, and the philosophy here is that even though a fairly small number of people are affected by these diseases, if we come up with a treatment, it may be possible to expand the use of this drug to a larger population. And so one example of this is our effort in spinal muscular atrophy. So SMA is the most common genetic cause of infant death. It's an autosomal recessive disorder that is associated with loss of, with mutations in the SMN1 gene. So SMN is survival of motor neurons. And so if you lose both copies, you get spinal muscular atrophy. Now, it turns out that there is a second gene that is identical called SMN2, And depending on the copies, so different people have different numbers of SMN2, and depending on the copies of SMN2, you get either more severe or less severe disease. Now, the interesting thing about SMN2 is that even though it's identical, it is very poorly spliced. And in fact, specifically exon 7 is poorly spliced, and so the majority of SMN2 protein, of SMN2 message, is missing exon 7 and therefore produces a protein that just gets degraded. So we developed a screen to look for things that would correct splicing. At a time, I should say, when people thought that RNA was not druggable. Okay? And so Rajiv Sivasankaran in the group developed this assay in which we could use luciferase to determine whether exon 7 had either been spliced in or spliced out. He screened a large library. He got some hits. These hits were then improved with medicinal chemistry. Actually, he screened 1 million compounds. He got exactly 2 hits. And it turned out that eventually one of these was improved to the point where it would actually increase the production of SMN in the brain of a mouse that was lacking SMN1. In fact, this is a mouse that had the human gene. And it would actually prevent these mice from dying. This is LMI. And just recently, we have started dosing the first set of babies. And so this is also unusual because we decided to take this drug into babies, into children, before we took it into adults, because these are the people who are most severely affected. Okay, let me tell you a little bit about some of the projects in psychiatry and neurodevelopment. So one of our areas of interest is in schizophrenia. We have a collaboration with the Stanley Center at the Broad Institute at MIT. They have been heavily involved in identifying common SNPs that are associated with schizophrenia, as well as doing resequencing to identify less common mutations in the same genes. And the question here is how to take something like this. So this is a Manhattan plot. Each one of these is a genomic region that is associated with increased risk of schizophrenia. And the question is how to take this and convert it into a drug. And so the answer is we don't really know. But we have been trying to see whether we can cluster these hits either into signaling pathways or perhaps into specific cell types. And so, for example, we know that there are many hits here that seem to impact genes and proteins that are important for synaptic function. So we have generated screenable models for some of the synaptic defects that we expect would be seen if you were missing or had reduced levels of some of these genes. The other thing we notice is that if you actually look at all these genes, they're in fact enriched in a particular pathway, in a particular circuit, which is this cortical striatal thalamic pathway. And in fact, many of these genes seem to be expressed in particular cell types. And so we have embarked on ways of trying to increase or decrease the activity of a cell type as a way of trying to generate a treatment that would work for people with a variety of risk alleles. Okay, let me tell you a little bit about the work we have been doing on neurodevelopmental diseases. And I'm going to start with something that I've been working on for a long time, something called Timothy syndrome. So Timothy syndrome is a fairly severe neurodevelopmental disease that is associated with continuous syndactyly hypoglycemia, autism, and cardiac arrhythmia. It was discovered actually by Mark Keating's group. Mark Keating is also at Novartis. They were not interested in autism at all. They were interested in cardiac arrhythmias. And when he sequenced the patients, he discovered that there was a mutation in this gene, CAV1.2, which is a voltage-gated calcium channel that is expressed both in the heart and in the brain. And the mutation is this glycine for arginine substitution at position 406. And what it does is it changes the kinetics of the channel. It turns out only one splice isoform of the channel. So the channel, instead of opening and closing, as you see here, so this is current as a function of time, and if you depolarize the cell, the channel opens and then it closes. Instead, it opens and it stays open. And so the question is, why does this cause autism, and what are the phenotypes in cells from these individuals? And then finally, if we develop a drug that can address this issue, might this be helpful to somebody else? So first of all, we had to identify a phenotype, and so we generated neurons from these kids. We used we developed a whole series of cellular assays. One of the assays is illustrated here. It's a dendritic arborization assay. And the reason we decided to look at dendritic arborization is that it was known that L-type channels are critically important for the formation of dendrites in the developing brain. And of course, the iPS-derived cells are in fact developing neurons. So what we did is we introduced these light-activated ion channels into the cells, and then we stimulated them to fire action potentials, and then we would take these images over the course of 12 hours. And then we have software that extracts every one of these extension and retraction events. And so, if you do this, you can see that under normal circumstances in control cells, so these are 7 control individuals, dendrites expand over time as you would expect. But if you have the Timothy mutation, and I should say we started stimulating here, as soon as you start stimulating the cells, you start getting retraction. And we've worked a lot on the underlying mechanism of this. We know, for example, that at least part of this effect is independent of calcium. We know that it depends at least partly on the ectopic recruitment of these Rho-related small G proteins. But one of the things we could do is we could actually use this as a way of developing a phenotypic assay. And we developed a high throughput screen that is sort of illustrated here, where these are sort of wild type cells. So if you sort of stimulate them with light, you get this increase in the dendritic arbor. These are the Timothy cells. The red dots and the blue dots are just different time points. And then we screened a large library. We got lots of things that made no difference at all, but then there were a few hits. And some of these are quite interesting. This one, for example, is one that's public. This is roscovitin. Now, what's interesting is that it was in the collection not because it does anything to L-type channels, but because it's a blocker of CDK5. It turns out that if you then just look at roscovitin derivatives, we can show that the capacity to reverse this is independent of CDK5. In fact, it turns out that this molecule binds to the mutant L-type channel and reverses the molecular defect. So it actually causes the L-type channel with the mutation to inactivate. So now this could be useful, not just simply for autism. Mark Keating is upstairs, so we continue working on cardiac arrhythmias. It turns out that if you actually make cardiomyocytes from these cells, you can also see a phenotype. This is important because we'd made the Timothy mouse and it had no cardiac phenotypes, possibly because the mouse heart beats at 600 beats per minute. So in cardiomyocytes from iPS cells and controls, you see beautiful beating that is nice and uniform. If you look at this from patients that have Timothy syndrome, right away you see that there's something wrong. The cardiomyocytes are beating, but they're beating very slowly, so they're kind of bradycardic. In addition to this, they're missing certain beats. And furthermore, if you actually then patch clamp the cells, you can see that the action potential is very prolonged, and that every so often they have these little events, which are these delayed after depolarizations, which we actually think actually underlie the ventricular fibrillation that is actually the really dangerous event that is associated with this kind of long QT syndrome. Okay, so I'm going to tell you about just a couple of other examples of things that we've worked on or we're working on. So another disease we've worked on is Phelan-McDermid syndrome. It's also called 22q13 deletion syndrome. And 22q13 deletion syndrome is caused by the deletion of generally a large number of genes in chromosome 23, in chromosome 22. And it's associated with sort of neonatal hypertonia, this global developmental delay, and absent or severely delayed speech. Now, in addition to this, so there were a bunch of questions when we started. So one of them, of course, is, given that most of the mutations delete a large number of genes, what is the gene that is important for the underlying phenotype? Now, there was a candidate already when we started, and the candidate was this protein called SHANK3, which happens to be in this region. And the reason for thinking that SHANK3 might be important is that people had found mutations in SHANK2 that were also associated with autism. And then there were some rare people with mutations in just SHANK3 that had some of the features of Phelan-McDermid syndrome. So SHANK3 is a synaptic scaffold protein that binds to a broad variety of proteins at the postsynaptic density. So one of the first things we did is we tried to develop an assay. And this turned out to be really hard. We wanted to be able to look at synaptic transmission, and we discovered quickly that, for example, if you looked at synapses in cells that we put in a well, it was very variable. whether you saw a phenotype or not really depended a lot on the density of the cells and the age and the health of the cells. So we eventually developed an assay by which we could have control cells and the Phelan-McDermid cells in the same dish at the same time, and we could record from them at the same time. And so when you did this, very quickly you notice that there was a very severe phenotype. Now remember, these are young developing neurons. But what you can see is that whereas control cells have beautiful AMPA receptor responses and beautiful NMDA receptor responses, the Phelan-McDermid cells have much smaller synaptic responses. And you can do this from hundreds of cells, and you can show that there are defects. So this is the current amplitude given the strength of the stimulus, right? And you can see that, of course, the stimulus gets bigger the more you stimulate, but it's much reduced in the patient cells. And this is true for both AMPA and NMDA receptors. In fact, so the question is why, right? Is this really due to just a SHANK3? So the first experiment was to see if we could rescue this by co-expressing SHANK3. And this is important because remember, this is a heterozygote mutation. And so in principle, you could generate a treatment by generating a drug that would just increase SHANK3 protein levels. And so we expressed SHANK3 from a cDNA, actually from a virus, a lentivirus. And you could see, for example, that in a mutant cell it would rescue synaptic transmission. It also rescued the evoked response. So you can see that this is now vastly increased. Now the one thing we noticed is that while it rescued the response, it didn't actually rescue it completely in the sense that there was some fraction of cells in which it was never rescued. And we don't really understand why that's the case. Now, the other thing we did is we decided to see whether there might be some way of trying to rescue this. And now these assays were way, way too painful at the time to actually do a real screen. And so instead, we decided to see whether we could actually look for things that people had already shown changed synaptic transmission. So we tried a whole series of growth factors. This is a small fragment, a small sample of them. And it turned out that one of them, IGF-1, actually rescued synaptic transmission in these Phelan-McDermid cells. Something is beeping. Okay, I'll let it beep. Okay, so we've worked extensively to try and determine how IGF-1 actually works. So you would have thought, right, that, remember, this is a heterozygous mutation of SHANK3. So we thought, well, maybe what IGF-1 does is it just increases SHANK3 expression from the other allele. So we did this experiment where we actually measured SHANK3 expression along with a marker for synapses called PSD95. And as you would expect, right, if you look at the controls, there are fewer synapses, and there's also less SHANK3. So we treated the cells with IGF-1, and to our surprise, we lost all the SHANK3 expression. So now you have almost no SHANK3, and you have, so there's almost no red here anymore, right? But now you have all this PSD95. So this was entirely unexpected. So what seems to happen then is that for some reason IGF-1 is reducing the amount of SHANK3 and causing an increase in the production of a different kind of synapse. So how could this be? Well, one of the things we noticed, so this was surprising because people used to think that every synapse had SHANK1, SHANK2, and SHANK3. And yet when we actually looked at individual synapses in human iPS-derived neurons, we noticed that there were many, many synapses that had SHANK3 and no PSD95, and many had PSD95 and no SHANK3. So it seemed as if the human neurons had a different complement of synapses than the rat neurons, all of which have, in fact, both. In fact, this is true not just simply in iPS cells. This is also true in human cortex. So again, there's this diversity of synapses. And so this may help to explain why it is that when we made the SHANK3 heterozygote mice, they have essentially no phenotype, but the patients have a very severe phenotype, possibly because they're missing one entire population of synapses. In fact, we have a model for what we think is happening. And what we think is that SHANK3 in humans seems to identify these early-born neurons. And when you add IGF-1, it is gradually downregulated and replaced, perhaps by other SHANK proteins and by PSD95. If you have Phelan-McDermid syndrome and you only have half the amount of SHANK3, you have fewer of these immature synapses. But for some reason, IGF-1 manages to get the few that you have to become mature synapses, which is why you can actually rescue the defect. So does this matter? did we make any progress? And so, sort of while we were doing this, Joe Buxbaum was working on a mouse model of Phelan-McDermid syndrome. He was looking at not the heterozygote but the homozygote mouse, and he also found that IGF-1 could rescue some of the defects, some of the synaptic defects in the mouse. And so when both papers were published, this encouraged Alexander Kolvesen in his group to actually try a small clinical trial, which they have now just recently published. And so what's interesting here is that this is they've looked at social withdrawal in these kids, and so it seems that IGF-1 has a small effect on social withdrawal. Now, there are many caveats. It's a really small trial. This is a pretty poor measure of outcome. But on the other hand, we thought it was sufficiently promising that it would be worth developing something that worked in that pathway. Now, the reason that, so IGF-1 itself is a poor drug because it doesn't get into your brain very well, it has poor pharmacokinetics, it has all kinds of side effects. And so we've been working on something that looks sort of like IGF-1, but that is much more effective and is likely to be much safer. And so this is one of our development compounds. Okay, let me tell you a little bit about another disorder we're working on. Again, another neurodevelopmental disorder. This is 22q11 deletion syndrome. It's a congenital heart disease. It's a disease associated with congenital heart disease, palate abnormalities, autoimmunity. Now, the reason we care about it is that 25% of these kids have autism and 25% have schizophrenia. It is actually the most common genetic cause of psychosis. And so we have been making iPS cells from individuals that have 22q11 deletion, and we have some preliminary results on some of the things that may be wrong. So the first thing to notice is that if you just simply make neurons from these individuals and you measure gene expression, there are a large number of genes that are misregulated, but a significant fraction of them are genes that are actually also identified by the genome-wide association study. So there is some overlap between some of the genes that are misregulated in 22q11 and some of the genes associated with broader schizophrenia. Now, in addition to this, we've also looked at some of the physiological properties of the cells. I would say the most interesting one is the defect in the activity of the voltage-gated calcium channel, the L-type calcium channel, because this is a channel that has been implicated in a number of neuropsychiatric diseases. And so, in fact, the cells from the 22q11 patients, and I think these are 7 individuals, and there are 7 controls, have a much smaller calcium elevation. And in fact, we've looked at this pretty carefully. It turns out that this is not due to a defect in the calcium channel kinetics or expression. It's primarily secondary to a defect in the potassium channel that is also decreased. So this is potentially interesting. I think what we find to be even more interesting is that if we treat the cells with a dopamine, a D2 antagonist, which is an antipsychotic, and these cells, of course, have D2 receptors, you can actually reverse some of these phenotypes. So this suggests that that there is some defect that we don't exactly know, what the defect is in the potassium channel, but there's some defect that can be reversed by antipsychotics, which suggests that this perhaps is a valid cellular model for psychosis in cells. So one final disease we've been working on. This is Dravet syndrome. Now, Dravet syndrome, as many of you may know, is an intractable early onset epilepsy. We're interested because it's not just epilepsy, it's also associated with pretty severe cognitive abnormalities, so autism and developmental delay, and it's caused by a heterozygous loss of function mutation in a sodium channel NAV1.1. Now, you would think, right, that a loss of a sodium channel, which is in fact the sodium channel that mediates the action potential, would not result in epilepsy, but nonetheless it does, and so the question is why. And there is a lot of controversy because the NAV1.1 mice, the heterozygotes, some of them seize and some of them don't seize, and that depends on the genetic background. And people had actually already made some iPS cells, and there were 2 groups, 3 papers. So one paper said, it seems as if there is a defect there is a defect in the excitability of inhibitory neurons, but that group didn't make the excitatory neurons, and the other group said, no, loss of NaV1.1 results in the expression of another sodium current, so another sodium channel that makes the cells more excitable. So if we were going to work on this, we needed a really reliable model, and so we set out to determine whether we could actually identify a reliable phenotype. So Yishan Sun, who's a member of the group at Novartis, generated these inhibitory interneurons as well as excitatory neurons. He developed a protocol by which he could generate both of these at once. And he developed markers that allowed us to look at the inhibitory neurons that were mature. And then he did some electrophysiology, some automated electrophysiology actually, and he measured the sodium currents, and it turned out that, in fact, the patients in inhibitory neurons had much smaller sodium currents, but the excitatory neurons had perfectly normal sodium currents. So this supported the idea that the epilepsy in these kids is due to somehow a selective effect on these inhibitory interneurons. In fact, when you actually look at these are fast spiking interneurons, so if you look at the spiking frequency in controls, they spike very quickly. If you look at the mutants, in fact, when you stimulate them, they get into this kind of depolarization block. And of course, you can actually see this is 3 healthy controls, and these are some Dravet patients with various classes of mutations. And again, you can see that as you stimulate, as you inject current, right, initially you see increased firing, increasing action potentials, but over time, in the Dravet patients, the number of action potentials decreases. So there's a defect in the input-output curve. Because we're a drug development company, we want to understand how, whether we could develop any treatments for this. Now, one thing that's already in clinical trials is this molecule, cannabidiol. It's a derivative of cannabis. And people have anecdotally reported that it can act as an anti-epileptic. And so we wanted to know how it might work and whether it's likely to reverse all of the defect in Dravet cells. So cannabidiol is quite interesting. So at therapeutic doses, so these are the inhibitory interneurons. So at therapeutic doses, it has very little effect on excitatory neurons. But in inhibitory neurons, it actually, interestingly, sort of increases the activity of inhibitory interneurons. Now, it doesn't do it over the whole frequency range, so it doesn't actually, so these are the patients without cannabidiol, these are with cannabidiol, and these are the controls. So this doesn't look like this. Nonetheless, it does partially rescues the phenotype. But this also tells us that this is unlikely to be a perfect treatment for this disease, which has sort of encouraged us to then develop our own compounds, and we now have some compounds that can rescue the electrical activity in these cells. Okay, just to finish, I just want to just touch a little bit on our final initiative, which has to do with trying to improve our clinical trials. Now, as I said before, we could make the best drugs in the world, and we are going to fail unless we come up with better ways of measuring outcomes. We're doing these trials where we're measuring, for example, cognition once a month, once every 6 months. And in fact, the data, not surprisingly, is unbelievably noisy. So what can we do to make things better? So to some extent, by going for genetically defined diseases, we're eliminating one of the problems, which is that there's a lot of diversity in the population. So we can actually stratify patients according to genetics and not the genetics and pathophysiology and not behavior. So this ought to, especially if we have a drug that's aimed at a mutation, this should help us reduce some of the noise, but it's not going to be enough. The second problem is basically we're measuring these endpoints episodically, and this is a mistake. This makes it very, very difficult to actually generate clean data, and so we are investing very heavily, essentially, in gadgetology. Ways of both passively and actively measuring outcomes over a prolonged period of time. And then finally, of course, the noise, contributes dramatically to what is kind of the bugaboo of neuroscience trials, which is the placebo effect, right? Now, one of the main reasons the placebo effect is so big is because the variability in these outcomes is so enormous, right? So we hope that by generating better outcomes, we'll be able to reduce the placebo effect. But at the same time, we're also implementing better clinical trials with things like, for example, run-in periods and parallel arms that should help us reduce some of the placebo effects. Okay, so let me just finish. I started with telling you why everybody's getting out of neuroscience. And, the question is, can we succeed? It is really, really hard. It is really difficult to develop new treatments, and it takes a long time, and it takes a lot of money. On the other hand, I don't think we have any choice. I think we have to succeed, because if we don't succeed, we are going to face, a million problems. Let me just finish by thanking the people who made all of this possible. I'm just the, how shall I put it, the tip of the spear here. I often say I. When I say I really mean we, and when I say we, I really mean them. And them is these people. This is the Novartis neuroscience group. Of course, we have help from all kinds of other people at Novartis, chemists and geneticists and clinical trial specialists and so forth. And of course, we also know that we can't do any of this alone. So we have established collaborations with many people, including, of course, NIH, but MIT, the Banner Alzheimer's Institute, Stanford, which is my former institution, and a number of small companies. So with that, thank you.
Thanks for a great talk. We have some time for some questions. I would remind people to please use the microphones in the aisles so that people watching by videocast can hear your questions.
Hi, thank you for a great talk. I was curious to get your thoughts about the reality of trialing drugs. Let's say you find a druggable target for a neurodevelopmental disorder. I suppose there are two kind of interventional scenarios or extremes. One is that it's a phenotype you can reverse in adulthood, and another is that the action is happening early in development, and that's what you need to modify. And if that is the case, how do you deal with that challenge of modifying a sort of uniquely, a developmental process that may have a unique time course in humans, and therefore having to face trialing a synapse-modifying drug in children?
Sure. So, we've started to pioneer this with our SMA trial, where we're starting in babies. I think it's, given how poor the outcomes are, I think it's not at all unreasonable to think that one would do a trial in developing children. I think the challenge actually is developing endpoints that are short enough. It's very challenging and very expensive to do a very long trial. So if we can develop endpoints that are sufficiently fast, I think it's perfectly reasonable to do a trial in children. I mean, like everything else in drug development, it's really a question of risk-benefit. And I think in many of these cases, if we show that there is a benefit, I don't know of any parent who would say no. I can tell you as a parent, right? So I think it's possible. When do we want to start? I think for neurodevelopmental diseases, we want to start as early as we can. Especially, I should say, if we're actually addressing the underlying molecular defect, right?
I have a quick question. Do you have any thoughts on some approaches to speed the use of iPS-based cell therapies into the clinic? My understanding is they are not currently ready.
Well, I mean, I think it depends very much on what disease you're interested in. I think the lowest hanging fruit there are going to be applications where it's, the cell that you want to make, you have the protocol for making it, and you're fairly certain that introducing that cell back into the brain is going to work. So, all things being equal, you're better off trying to, for example, treat Parkinson's disease by introducing cells that secrete DOPA or dopamine, right? Because you already know that L-DOPA works, right? And that has some chance of succeeding. On the other hand, trying to treat autism by introducing iPS-derived neurons seems, crazy. Nonetheless, this does not stop people from claiming that they can do this in advertising online, but
okay, off to the right. Question?
Yeah, I was just curious to know about the drug which you were trying for the neurodegenerative disease, the Alzheimer's. You mentioned something about a drug where you were trying for the targeting the A beta and the tau. And can you specify which drug you were trying, I mean, or at what stage it is?
Yeah, I can't.
Okay, that's okay.
This is a little embarrassing. I mean, I think, like many other, I should say, many more pharmaceutical companies are leaving psychiatry than their neurodegeneration. So there are, quite a lot of companies working on neurodegeneration, and I think everybody's thinking about what they can do about tau. I mean, I think the underlying hypothesis of why it is that tau is toxic and what it does is not so clear. There seems to be reasonable evidence that it spreads, and so there are a whole bunch of anti-tau antibodies that people are testing, and we have a whole take on how we might try to interfere with tau secretion and spreading using small molecules. But it's early. We'll see if it works.
Okay, one final question, please.
Hi, Dr. Dolmetsch. My name is Omar. I'm a medical student. I loved your presentation. My question is, so recently in Nature they were talking about how the lymphatic system is actually going further higher than the cervical area into the dural sacs of the brain, having connections there. I was wondering what you thought about the implications of that for like neural pharmaceutical compounds.
Yeah, it's a good question. In general, not just in the CNS, we are not very good at addressing any problems in the lymphatics, period. I don't think we really know what the lymphatic system is doing, in the CNS. So I think it's a little early, but I think it's very exciting and very promising. It seems, for example, that there are at least some subsets of neurodegenerative diseases where eliminating misfolded proteins and other sorts of waste are limiting, and perhaps one can harness the lymphatics that way. But I should say, we sort of suck at doing this in every system. So, but it's a very interesting finding.
Okay, please join us again for FAES-sponsored reception in the library. Thank you.